I welcome you all in the plant physiology and metabolism course. Today I am going to talk on the fatty acids. The fatty acids play a fundamental role as the building blocks of lipids, forming essential structural components within these biological molecules. Originating as derivatives of hydrocarbons, fatty acids are composed of a hydrocarbon chain and a carboxylic group. Generally, fatty acids exhibit an even number of carbon atoms spanning a range from 4 to 36. The most prevalent fatty acids typically consist of 12 to 24 carbon atoms. The hydrocarbon chain in fatty acids may adopt either a branched or unbranched configuration, with the majority of common fatty acids featuring unbranched hydrocarbon chains. This diversity in fatty acid structure underscores their versatility in contributing to the varied functions of lipids in biological system. Fatty acids exhibit a dichotomy between being saturated or unsaturated, determined by the presence of double bonds in their hydrocarbon chain. The defining characteristics of unsaturated fatty acids lie in the number and positions of these double bonds. In the case of unsaturated fatty acids with double bonds, the arrangement involves two consecutive double bonds separated by a methylene group. A straightforward system for abbreviating fatty acid structures utilizes a two-digit code separated by a colon. The initial digit signifies the number of carbon atoms in the hydrocarbon chain, while the subsequent digit denotes the count of double bonds. For instance, the abbreviation 16:0 correspond to palmitic acid, signifying a 16 carbon hydrocarbon chain devoid of double bonds. Similarly, stearic acid is abbreviated as 18:1, indicating an 18 carbon hydrocarbon chain with a single double bond. To specify the position of the double bond, delta nomenclature may be employed. For instance, stearic acid could be represented as 18:1 delta 9, pinpointing the location of the double bond between the ninth and 10th carbon atoms. In cases with two double bond between the ninth and 10th carbon atoms, the notation 18:2 signifies the structure of linoleic acid. This systematic approach to representing fatty acids provides a concise and informative means of conveying their structural attributes. This table displays the structure of some of the common fatty acids found in the plant. That here if you can just see in the table, the saturated fatty acids are the lauric acid, and it is donated by 12:0, and then we have a myristic acid donated by 14:0, then palmitic acid donated by 16:0, and stearic acid donated by 18:0. Whereas in the unsaturated fatty acids, the oleic acid is denoted by 18:1, linoleic acid is denoted by 18:2, and linolenic acid is denoted by 18:3. So, this slide shows about the fats and oil store, which has been large amounts of energy. So, fats and oils belonging to the broader category of lipids constitute a structurally diverse group of hydrophobic compounds. In terms of carbon content, lipids represent a more reduced form compared to carbohydrate. Their unique characteristics lies in being soluble in organic solvents while exhibiting high insolubility in water. An intriguing aspect of lipids is their capacity to yield more ATP, that is energy, through complete oxidation compared to carbohydrate. Specifically, 1 gram of fats or oils can produce around 40 kilojoule of energy, a substantially higher value than the 15.9 kilogram of energy obtained from the oxidation of 1 gram of starch. It is noteworthy that the biosynthesis of lipids demands a considerable investment of metabolic energy. This intricate interplay of solubility, energy yield, and metabolic investment underscores the pivotal role lipids play in cellular processes and energy dynamics within the biological system. Moreover, beyond their role as energy storage, lipids are indispensable for various aspects of plant structure and functions. Particularly, phospholipids and galactolipids, that is the integral component of plant membrane, contribute significantly to membrane integrity and functionality. Sphingolipids, that is the another class of lipid, play a crucial roles as important membrane components contributing to the dynamic composition of cellular membranes. Then the waxes serve a protective function by forming the cuticle, effectively reducing water loss from exposed plant tissues and providing a shield against environmental stresses. Additionally, terpenoids, also known as isoprenoids, constitute a diverse group that includes carotenoids, which is essential for photosynthesis, and sterols, which are present in many plant membranes. The multifaceted roles of these lipids underscore their significance in supporting the structural integrity and functional processes vital for plant growth, development, and adaptation to the environment. This figure basically illustrates the structural features and polar glycerolipids in higher plant. Fats and oils predominantly exist in the form of triacylglycerols, with the term acyl referring to the fatty acid portion. In triacylglycerol, fatty acid molecules are connected through ester bonds to the 3 hydroxyl groups of glycerol, forming a crucial structure for energy storage in organisms. Plant derived fatty acids, a major component of these triacylglycerols, typically comprise straight chain carboxylic acids featuring an even number of carbon atoms. The length of these carbon chain varies, ranging from as few as 12 units to over 30. However, the most prevalent length observed in plant fatty acids are 16 or 18 carbons. This diversity in carbon chain length contributes to the versatility of fats and oil, enabling them to fulfill essential roles in energy storage and various physiological processes within organisms. As you can see in the figure, the triacylglycerols, commonly known as triglycerides, fats, or neutral fats, represent the most basic form of lipids constructed from fatty acids. These molecules consist of 3 fatty acids, each linked through an ester bond to the single glycerol, forming a fundamental structural arrangement. As you can see here in the figure, a cross section of human white adipose tissue. In vertebrates, a specialized cell called adipocytes or fat cells store large amounts of triglycerides as fat droplets that nearly fill the cell. Triacylglycerols are also stored as oils in the seeds of many types of plants, providing energy and biosynthetic precursors during seed germination, as you can see in the figure B, that is the cross section of cotyledon cell from a seed of Arabidopsis. The physical state of oils remaining liquid at room temperature can be attributed primarily to the prevalence of carbon-carbon double bonds, that is unsaturation, in the fatty acid constituting them. In contrast, fats characterized by a higher proportion of saturated fatty acids, exhibit a solid state under similar conditions. The composition of fatty acids within plant lipids varies across plant species, exemplified by the distinctive makeup of oils like peanut oil and cottonseed oil. Peanut oil, for instance, contains around 9% palmitic acid, 59% oleic acid, and 21% linoleic acid, while cottonseed oil comprises 25% palmitic acid, 15% oleic acid, and 55% linoleic acid. The storage of lipids within plant cells is facilitated by organelles termed oil bodies, alternatively known as spherosomes or oleosomes. These structures are located in either cotyledon or endosperm cells. The oil body membrane is characterized by a single layer of phospholipids forming a half bilayer configuration. In this arrangement, the hydrophilic ends of the phospholipids interface with the cytosol, while the hydrophobic acyl hydrocarbon chains orient themselves toward the interior where triacylglycerols are stored. This intricately structured system supports the storage and regulation of lipids within plant cells. Oleosense are specialized proteins found on the outer surface of oil bodies, play a pivotal role in ensuring the stability of these structures by preventing the interaction and fusion of phospholipids with neighboring oil bodies. The distinct composition of the membrane enveloping oil bodies arises from the specific pattern observed in the biosynthesis of triacylglycerol. Enzymes responsible for triacylglycerol synthesis are located within the membranes of the endoplasmic reticulum. Throughout this synthesis, fats produced accumulate between the two monolayers of the endoplasmic reticulum membrane bilayer. As additional fats are continually integrated into the structure, the bilayer expands, culminating in the formation of a mature oil body that separates from the endoplasmic reticulum. This intricate process underscores the significance of oleosense and the dynamic interplay involved in triacylglycerol synthesis, contributing to the nuanced structure and functionality of oil bodies within the cellular framework. This is the table showing the structure, properties and nomenclature of some of the naturally occurring fatty acids. So, thank you. In, in the next session, we will discuss about storage lipids.